Alright, in the last video we talked about how splicing can alter the resulting protein made. Well, now we're going to go and we're talking about how certain mutations can alter what we call the reading frame. So what is the reading frame? Well, we're going to remember we had our double-stranded DNA, and we copy one of those strands in the form of an mRNA. So here is our DNA, and we have RNA, and then we go to the ribosome, and the ribosome turns that into an amino acid sequence. I'm going to draw the amino acids kind of like this, and then I'm going to draw a line through them. So this is our amino acid sequence. Sometimes you might call that a polypeptide. I'm going to abbreviate it here. All right, and then when it folds up on itself, this thing will kind of get wadded up, you know, like a string that gets wadded up into a ball. Well, we call that folding, and when you fold this thing up, you call that a protein, functional protein here. Well, the thing is, this sequence up here are the instructions for making this. Anything that alters the sequence of letters is going to alter the protein that you get. So we're going to talk about how some mutations sometimes can alter this, and it all has to do with letters. So we're going to start, and I'm going to make a fake protein here. We're going to pretend that our protein has 4 amino acids in it. We're going to make them all the same. We're going to make them all methionine. All right, MET, methionine. And it's going to be bound to another methionine. Okay, and methionine. And methionine. All right, so we've got 4 methionines in our protein. We're going to go backwards, though. We want to go backwards and get to the DNA. But to get to the DNA, we have to go to the RNA. What RNA coded for this protein here? Well, the RNA that coded for this, we know that there's only one codon that codes for methionine, and that is AUG. So we know that the RNA that made these 4 amino acids is a sequence of letters AUG, 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 AUG. So this is our amino acid sequence. All right. Here is our RNA. We, of course, know that it's RNA because it has U's in it. Now we need to reverse transcribe, I guess, back to the DNA that produced this. Well, the DNA that produced this, we have to use the genetic code, and not the genetic code. That's going here to here. The genetic code, AUG, codes for methionine. The genetic code is what you use here. But here we need to find the DNA complement to this. So the DNA complement, if this is A, then what's going to be here? It's T. We're going to DNA here. We're going kind of in reverse in the way you normally think of it. And if this is a U, this must be an A. If this is a G, this must be a C. So this is the DNA that produces AUG. So we're going to go tack, tack, tack. So this is the DNA that produces this. All right. Alright, so RNA polymerase will attach to this, make that, and then you go on to make your amino acid sequence, that will fold into a protein. So if you notice, it reads them what? 3 at a time. It reads these 3, these 3, these 3. Alright, or if you want to think of it up here, it actually, I guess the ribosome you would think reads these 3, then these 3, and they're read in this order because AUG codes for methionine, so it's going to read them as a codon. Codon is just 3 nucleotides, all right, in a row, and you're going to read these as triplets, all right, as a codon, all right. And so the DNA that made this, if this DNA is changed, we're going to change this, And then, of course, that's going to change our protein. We don't want that, or if we do, we don't want there to be a huge change. Hopefully one letter change. If I change this from an A to something else, well, okay, maybe it just changes one of the methionines. But there's one particular type of mutation that's really bad. It changes the protein a lot, and that's called a frame shift. And a frame shift is usually an insertion or a deletion. What happens if suddenly I just take one of these out, or if I add one in? Well, that's going to cause what's called a frame shift. So we say that insertions or deletions cause frame shifts. Oops. And in this particular case, this is our frame. And when they say this is our frame, it means I'm reading these three, followed by these three, followed by these three. If I were to get a frame shift, then I would be reading these three, followed by these three, followed by these three. You don't know what this one is, and you don't know what these two are, but they would be part of this frame. So you have three possible frames. All right, you could be the one that we want, which is the AUG, 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 but you could also have one that is UGA, UGA, UGA. And then, of course, the third one would be if you're reading it GAU, GAU, GAU. Okay, so those are the three frames. Well, let's get another picture here. All right. So if we are going to be doing this and we have an insertion or a deletion, so we would normally think of it as our methionine, 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 and underneath that we have AUG, 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 and then the DNA that transcribes for that is TAC, 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 TAC. So what happens if we have an insertion or deletion and suddenly we're reading this in a different frame? So we might call this frame 1. All right, so frame 2 would be, okay, we're starting here. Frame 2 we're going to move over. Our DNA, remember this is the DNA here, it's where we start. So if suddenly, let's say this T were deleted, we would now be our DNA sequence, if that were deleted, would now be ACT, ACT, AC, and we don't know about that one, so we'll just start here. All right, we had that deletion and now we're reading ACT. Well, ACT does not code for the same thing. All right, and let's figure out what it would do. So ACT codes, if we translate this into RNA, this would be what? UGA, UGA, and UG something. All right, we don't know on that one. We had that deletion there. Actually, I left one out. We'd have another AUG there. But what is AUG if it's translated by the ribosome? This is now what goes to the ribosome. And what happened if we had a frame shift? If we had a deletion and it shifted the frame, we, over here, we'll call this frame 2. If we shifted the frame, we now are not coding for a methionine. We're coding for, this is a stop codon. Very bad. That means if you get this mutation, you simply stop making amino acids. Okay, you've told the ribosome to stop here, and that's the end of your protein. So you have a premature end to your amino acid sequence, which is more than likely going to alter the functionality of your protein. So very bad if you had an insertion and deletion. You may also have what's two deletions or maybe an insertion, and you might go also to frame 3. So what would frame 3 look like if we go just one step more? And what I mean by that is, what if we were starting here at instead of what, ACT, ACT, all right, what if we started with the C and go CTA? So we would get CTA, CTA, CTA. So let's see here, CTA, 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 one more C there, but we don't know what the last two are down here. So this would be our new reading frame. We call these reading frames. So the parts that get read. Let's translate this though. Transcribe this. So a CTA goes to a GAU in the RNA. GAU. We don't know about this one. It's a G, but we don't know what the last two are. But in this new reading frame, after the shift, that's going to code for the amino acid aspartic acid, ASP. Now, you might ask, well, which is worse? Well, here you prematurely stopped making your protein. May make it where it's not functional at all. Don't know. But it is being made. Over here, you're adding on new amino acids. This may just simply make this not work. You were supposed to get methionines, and you're getting aspartic acid. But it may change its function. This may be poisonous to the cell. It's hard to say sometimes which of these is worse. Here, the protein just may not function. But here, or maybe you just stop making it here or something. But here, you might change the chemical properties so that it actually is harmful. Not making it sometimes is preferable to what? To making something that's poisonous. Here, remember, you've got two copies, so maybe if just this one doesn't work, that's fine. You have the other copy on the other chromosome, so maybe it doesn't kill the cell, so this might not be too bad. But over here, now you're making a protein, then you don't know what it does. It might have different chemical properties, which might be poisonous in the sense of toxic to the cell. All right, so these are frame shifts, and I wanted to show you what they were and why it's important to understand what the reading frame is and what frame shifts are, and if you are in different frames. And if you go negative, all right, so there can be a negative frame, all right, so that means you're reading it backwards, all right, you're going in the other direction and reading it in that direction instead of this direction. And you're going to see that in the worksheets. They're going to have a negative frame, so just keep a lookout for the negatives.